Hello, hi. Today we will continue how we can develop biofertilizer and also microbial inoculants. The six mechanism that I mentioned just now. Yeah, so in the exam, if we say what are the beneficiary property that can be brought about by biofertilizer, so you have to elaborate the six like what I did now. Um, some of the microorganisms also have the ability to suppress the growth of pathogen. We call it phytopathogen, yeah, because phyto means plant, pathogen that attack plant. We call it biopesticide. These are biomolecule that can exert kind of biotoxicity or prevent the phytopathogen from growing. Sometimes the MI is also closely related to phytostimulators or bioenhancer due to the phytohormone, like what I mentioned just now, IAA and AIA. You can get the full term from the website easily. We call it phytohormones, which will enhance the plant growth. Yeah. Another one is called rhizoremediators. Once you see rhizo, means that these are a group of microorganisms that can promote the growth of roots, plant growth. When we say remediator, meaning that it can promote the degradation of organic pollutants, especially at the root part plant. Okay, so why do we care about microbial inoculant? These are the type of biofertilizer that can promote the biological active products. It can be in the form of compost. Typically, it contains one or more beneficiary bacteria and of fungal strain. You know the fungal because it has a lot of hyphae, okay? The fungi will have the hyphae, hyphae, like hairy structures to go through the solid soil efficiently, and so that it can work to segregate the enzyme through the hairy structure of the hyphae. This can improve the nitrogen fixing and phosphate solubilizing. Also, other enzymes like cellulolytic cellulolytic actions, they can promote the plant growth and yield by improving the availability of soil nutrient in its rhizosphere. As I mentioned, this is the area around the root, hairy roots and so on. Typically, any MI product, microbial inoculant, will contain two microbes and added with a carrier material to stabilize the MI within the carrier material. Most popular microbe includes bacteria, blue-green algae, and arbuscular mycorrhizal fungi, or in short we call it as AMF, very commonly used as a MI for agriculture. And then there is this microbial biopesticide. The best well-known strain is called Bacillus thuringiensis (BT). If you Google from website, you can see a large informations where it will provide a kind of enzyme, release a kind of enzyme when go into the caterpillar. It will cause the caterpillar disrupt the digestion system of the caterpillar, and the caterpillar will refuse to eat the leaf, then die of hunger. Yeah, quite cruel, yeah, but this is the genetically modified BT, largely produced in the US. So the advantage is that all these MI has the advantage of ease of application, eco-friendly, cost-effective, and non-toxicity to human being. Although it will be toxic to the certain pests such as the worm or caterpillar. So there are increased awareness of the public towards health and ecologically friendly selections of biopesticide, so that we can reduce the use of synthetic chemical pesticides. More justification for the use of MI in the bioproducts such as biofertilizer and biopesticide. I don't have to read, you can read it yourself. Quite similar to the 6 major property that I presented in the earlier slides. Yeah, nutrient solubilizations, higher pathogenic effect, soil fertility. As you know, a lot of these phosphate and nitrogen, they are not readily solubilized in soil. That's why we need the microbe to do the work. As you know, NPK are the major macronutrient to grow any plants. Nitrogens mainly to grow is a common compound to synthesize chlorophyll to enable photosynthesis to happen. So this is universal, needed for all the vegetations and the productions of leaf and the overall growth of the plant, and to determine the yield of the crop. And also all these nutrients are needed in the larger amount. That's why it's called macronutrient, to synthesize secondary metabolite to promote plant growth. These are usually hormone or antibiotics, enzymes, phytohormones like auxins. Yeah, just now I mentioned the IAA and AIA are auxin group of growth hormone. Gibberellins and cytokines are all promoting plant growth. So as you can see, in order to produce the NPK in the chemical fertilizer plant, it will need a lot of energy per kg of the N or P or K. So this is talking about the kilowatt hour. If we do composting of the organic waste, we can reduce these energy consumptions worldwide, and hence it can also reduce the greenhouse gas emissions due to energy. Usually after formulated, right, the microbe will not die. The microbe is like in a very low metabolism rate. They need a very low oxygen, but they will, for example, if you go with some carbohydrate, it will have some small pore for oxygen to exchange, to sustain a very low oxygen into the capsulated microbe inside during hibernation. Yeah, so it will be in the hibernation state. And then they are, as I said, solid carrier formulation or liquid formulation. Of course, it can be entrapped in the polymer, or fluidized bed dry formulations using powder to fluidize the bed. But in our subject, we mainly focus on solid and liquid formulation. For solid, it can be in granules. Yeah, you can see that, for example, the animal feed, it has a biji like that, the bits like that. Or if it is smaller, it can be micro granules less than 1 mm. It can be wettable powders, meaning that it is in a powder form, but it can easily get wet. Wettable, water dispersible. When put into the water, it can directly disperse easily. It's called wettable or water dispersible granules. Powder is more fine. Granule is like a pellet like that. Okay, dust in the dust form or pellet form. Okay, all of this can be quite similar. It's just depending on the size range. You can just mention a few examples. In the liquid form, it can be in the suspension concentrates, or in oil, miscible, flowable concentrates, or ultra low volume suspension, or oil dispersion. And so mainly they will be in like an emulsion, oil in water or water in oil form. One spray, it can be easily, the good thing of liquid is that it can be easily sprayed and into a more fine droplets, and it can be easily stick on the plant, as compared to solid. The microbe may not be as active as in the liquid form. Yeah, okay. There are a range of material that can be used as a carrier. For example, soil base such as peat, coal, clay, lignite, and so on. Plant waste material such as compost can also serve as a carrier for the bacteria to attach. inert material including vermiculite, perlite, bentonite. Bentonite is quite a common material. It looks like the clay. Arginate, bead, and polyacrylamide are types of gel. Plant lyophilized microbial culture and oil-dried bacteria. Lyophilized means we make it into a powder under very low temperature, for example, freeze drying. Okay, or we can put some oil to coat the bacteria and dry it under low temperature, for example, 4 degrees Celsius, which enable the microbe to be incorporated into a soil, into a solid carrier, or to be used readily. Okay. Liquid inoculant containing some additive to improve the property of final products. Capsule-based carrier, so it's like cell will be encapsulated in capsules or in pelleted spore that the microbe will be hibernated inside the capsule or pore, and once we need to use, we can dispense in water for spraying and application to plant. But this kind of to compare the organic and inorganic carrier, for example, compost, manure, organic carrier, for example, clay, peat, coal, lignite, these are inorganic. Okay, the disadvantage of using carrier is that the shelf life is usually quite low, where the microbes are also sensitive to the temperature change. For example, suddenly too hot or too cold, the microbe might die. And there is also high risk of contamination. So when we apply the microbial inoculant, MI, the cell count is actually quite low. So relatively, liquid formulation will be a better alternative to the solid. These are all solid carrier in terms of formulating the biofertilizer. So this compare the advantages, highlighted the advantages and limitation of liquid fertilizer. So this can be in the aqueous suspension. It can be just a broth culture added with mineral oils or organic oil. As I mentioned, it is like oil in water suspension. Typically, this will have about 10 to 40% of the microbial inoculant in the liquid biofertilizer as a MI product, and then added with about 1 to 3% of suspended ingredient. This is like a surfactant which make the microbe to be suspended evenly, and also dispersant. Dispersant is also a kind of surfactant to increase the even dispersion of the microbe, and also surfactant. So these are all similar chemical, suspender, dispenser, surfactant. It's just that so when you call it as surfactant, it will really break the water and oil interface and become more miscible. But dispenser is more, suspender and dispenser are more like you can still see the microbe in the water droplet, maybe immersed in the oil. So this can be oil in water or water in oil suspensions. Okay, then to carry the micro inoculant is called carrier liquid. So this can be water or oil, depending on whether the micro inoculant is more having a longer shelf life, water or oil. So this will be decide the carrier liquid, which is majority 35 to 65%. The advantage for liquid based biofertilizer is longer shelf, lower risk of contamination, because usually all this carrier will be sterile first before mixed with the MI, which is already in sterile or in pure conditions. It will be easier to handle and apply because as I said, for liquid we can use the piping system to the fertigation setup to just spray and apply. But for the one with solid, it will require manual dispersing or larger hose to dispense. But the solid one still need to dissolve in water first. At the same time, this kind of so-called MI as a biofertilizer, we can also add other ingredients to improve the growth of the MI. For example, the low concentration of nutrient and also specific growth hormone to make sure the MI is sustained. But usually this will add extra cost. Okay? For the limitation, lack of solid carrier protection, lost viability after inoculations. In order to improve the shelf life of liquid-based biofertilizer, a range of additives such as sucrose, glycerol, gum arabic, polyvinyl pyrrolidone, or in short PVP, can be added to improve the survival of the MI upon application in soil. Also, this can enhance the inactivation of the toxic substance in the soil. This will, since this kind of carbohydrate-based additives are quite sticky, it can also improve the adhesion of the MI to the seed and stabilize seedling. Okay, enhance the survival of MI during storage and under various environments. For example, RP, Rhodosudomonas palustri, is a kind of photosynthetic bacteria. Strength pH3 could improve soil quality and increase the Chinese cabbage yield up to 40% as compared to the control. This bacterium can also detoxify pollutants such as hydrogen sulfide in a paddy rhizosphere. Yeah, so some of the compost will release hydrogen sulfide due to organic matter degradation, and this kind of smelly gas also need bacteria to detoxify it. It can also be a toxic to the paddy plant. And during the liquid formulation, there are 4 major parameters that we will need to control. Number 1 is this is usually the most common format as compared to the vegetable powder. As in the liquid form, it is absent in the dust and is to use and with high effectiveness. But in this case, we have to control the concentration of the suspension. So our biomass, the microbe, is considered as solid active ingredient. It has to be dispersed in water. Then after that, it can facilitate the dilution with water before application. So this is called suspension concentrate. So the biomass will be quite high. Another one is called ultra low volume suspension. This is ready to be used by ultra low volume equipment. For example, we want to generate very fine, extremely fine spray using aerial or ground equipment. We can use this ultra low volume suspension. Another one is oil miscible. Miscible is whether it can mix or not. Oil miscible flowable, meaning that this is oil and water mixture, whether it can easily flow. It's called flowable concentrate. So this suspension contains active ingredient of the MI dispersed in a solid. Also same with this one, it will need an organic liquid before use, because oil is soluble only in organic liquid for dispersion. Then it need pre-dilution in the organic liquid before spray to the plant. Okay, so this is special case when the microbe prefer oil interface, but usually is water soluble phase. Another one is oil dispersion, where active ingredient, meaning the MI, suspended in water, immiscible solvent or oil. Okay, this is miscible, this is immiscible. So immiscible means they are two-phase. They are not really mixed together to become emulsion. This will have lower evaporation rate and can remain longer with target plants, mainly because it is protected by the oil layer as a solvent to protect the MI. So example of questions, you have to describe and compare different formulation for liquid phase biofertilizer. 1, 2, 3, 4. Okay. There are more advanced formulations. For example, immobilization, meaning that the microbe will be captured inside some carrier to form flocculation. Is a kind of gumpalan, where the microbe will adhere to become a small ball of agglomeration, flocculation, or it can absorb on certain surface, either through weak van der Waals attraction or covalent binding. Yeah, but if when you learn about immobilized enzyme, if you have covalent binding, yes, it will adhere to the carrier stronger, but it will lose some of the activity within the cell. The activity mainly come from enzyme, hormone, and so on. So we can also do cross-linking of cell. Encapsulation is like you have capsule, your microbe put inside the capsule to enhance the efficacy of liquid biofertilizer by using polymer gel as protection layer. Okay, so the potential question is that describe how encapsulation enhance the efficiency of liquid biofertilizer. What type of polymer? You can compare natural and synthetic. Natural is, for example, sucrose and so on. Synthetic, it can be PVP, the polymer base, are often used for encapsulation, and each of them will have the pro and con. I have provided a journal paper for your further reading. And another potential question will be, what are the criteria of the polymer to be considered to be used as an encapsulation shell for the MI? Example answer will be, we have to consider the chemical composition, the molecular weight, and ability to interact with other compounds. Most importantly, the diffusion rate of nutrient through the encapsulation should be taken into account, because you have to consider some nutrient has to flow inside the capsule to give the microbe minimum nutrient to stay hibernated but not so active. So you have to control the chemical composition, the permeability, the diffusion rate of the nutrient. So these are the example of the answer, but of course you can quote more other. Okay, how plant biofilm serve as the biofertilizer? I think this is the carrier. Biofilm is a complex structure made up of microbial cells embedded in the self-produced matrix, which exert adhesion both to biotic and abiotic surfaces and help microorganisms to alleviate the adverse environmental conditions. So you elaborate on one example. For example, you refer to the journal paper given. You can elaborate there are, for example, 3 main types of biofilm may occur in the soil. Okay, so this biofilm is actually formed by the biomass cell itself. So when the cell attached to some surfaces, they become a film. This is called biofilm. So make up of biomass, the microorganism cell itself. So this cell can be bacterial, fungal bacteria interaction mixture, where the major part of the microorganism associated with plant roots are capable of forming biofilm. So they form this biofilm on the plant's root, and this structure will exhibit plant growth promoting property, mainly because when more microbes are adhered to the root, they will give some enzyme to mainly degrade some of the organic matter, so they enhance the nutrient release and uptake by the plant. This have been widely reported for a range of crop. Okay, so the mechanism is quite complicated. You don't have to specify what mechanism for what crop, but basically you should be able to describe this kind of biofilm based microbe living attached, absorbed on the root and releasing to form the biofilm and releasing a range of plant promoting metabolite. Yeah, that's why causing the plant to grow better than the plant which are not inoculated with the MI. Okay. Another potential question is how nanoparticles such as titanium oxide can be used to formulate liquid biofertilizer. So this is a good example of inorganic carrier. So again, you refer to the journal. Example of the answer you can extract from the journal further reading, including nutrient can be released in a controlled and gradual way. Yeah? So either slow or quick way through the emulsion formed with high surface area under the influence of different environmental factors such as temperature, moisture, and acidity or alkalinity of the soil, so that it will has enhanced viability and activity even after 12 weeks, 4 months of storage at room temperature. So sometimes you have to consider about the temperature factor in the warm weather or cold weather, the moisture percentage, whether is 40% or more than 40%. The soil alkalinity and acidity will also affect the solubility of your nanoparticle. So when you design the layer of the nanoparticle, you take into consideration of this physical condition in the soil. It will affect the release of the MI into the soil, so that the microbe will sustain good activity as well as not release too fast or too slow. Okay, and this nanoparticle can be used as an emulsion to coat the microorganism, okay, with high surface area. All right. Talking about how do we produce a biofertilizer, particularly rich in MI. Firstly, we have to select a suitable culture. Culture means a suitable microorganism, either 1 or 2 or 3. So it can be pure culture or it can be a mixed culture or complex culture. And firstly, we have to isolate the effective microbe. Usually, this will involve the work of microbiologists. They will isolate from the nature. Sometimes it can be from the forest, from the sea. If they get it from the forest, they will try to find some rotten root, rotten tree, because on the rotten root, they will have, it will be rich in fibers degrading microbe. From there, they will grow it in a petri dish that can isolate only those with high fiber degrading microbe. Is very good for composting, for example, but for soil, for other purposes, perhaps they have to isolate from different source. Okay, effective microbe is a general term to describe any microbe that can do effective work that will give beneficiary effect to the soil fertility or plant growth. Okay, and then after we isolate in the colony, in the selective agar, we have to determine the property of the selected microorganism on a proper medium with the appropriate growth conditions. So we have to put the right nutrient to grow it. And proper medium means usually refer to selective medium. For example, if you want to select microbe very good in degrading fat or protein or carbohydrate, you can choose a specific medium, so that only those, for example, you want to remediate oil, yeah? So you can put the medium only with oil, so only those can eat oil will be able to survive on your selective medium. And after that, we decide the growth condition, what are the suitable carbon source or nitrogen source needed to grow. We will do upscaling in a larger fermenter. So this one, usually we do it in a small shaped flask and the petri dish with selected medium. And then upscaling is usually up to 200 or 500 liter and above. After we have upscaled it, we have produced high density of biomass. This can be bacteria, fungal, and so on. We will select a right carrier. Remember, it can be organic or inorganic, remember? And then after we attach the microbe in the carrier or encapsulation or immobilization, then we formulate. Remember the formulation, the microbe is about 30% to 40%, and you have to mix with oil or water, dispersant, surfactant, suspendant, yeah, to make sure that it is stabilized and well dispersed in the carrier liquid. Yeah. After that, we have to apply in the field. Either we do it in a greenhouse or so that it will be in an enclosed situation. We can compare the performance of the plant growth in the greenhouse with and without the application of the MI. Or if you are more daring, we want to test it on the open field. But usually we do it in a greenhouse, comparing the one with spray with MI or without spray in the MI to validate the efficiency in increasing the macro, micro, and micronutrient in the soil, or in terms of plant growth, because some of the microbial inoculant will give a better plant yield than soil fertility. So there are a wide range of the MI doing different work. Or after that, we are sure what kind of concentration of the MI and the right formulation, we will further test in the open field. And the advantage of doing it in the greenhouse is mainly lack of ecotoxicological effect. Okay, so we can prevent, for example, some of the formulation or some of the microbe can cause ecology, maybe will make some beneficiary microbe or naturally occurring to loss, to extinct. Yeah, so this is called ecotoxicological effect. For example, when we inoculate microbe A, maybe the other beneficiary microbe like B, C, and D, which originally exist in the field, will die. This is called ecotoxicology. So we can test this, and then we can also evaluate the beneficiary impact in terms of plant growth promotion, and also increasing crop yield. Promoting plant growth, this mainly is contributed by soil fertility. Sometimes the soil fertility is good, but the yield may not be significant. It can also be the other way around. Maybe the soil more or less the same, but there are maybe secondary metabolite that can boost the growth yield as compared to the control, which is not inoculated with microbial inoculation. And then finally, once all this has been successful, there is no significant ecotoxicology effect, and there is really the extra beneficiary impact as control to the plant, as compared to the control. Then we will start to register the MI under a patent or copyright to the regulatory body for approval as a biofertilizer. Okay, so this can refer to one pure culture or mixed culture. Then once this has been approved, we will do further upscaling to 1,000 liter and so on, and produce at the industrial scale. This one upscaling here is only pilot scale, maybe around 500 liter, around there. Okay, industrial scale is usually 1,000 liter and above. Okay, and in doing so, we have to do quality control in terms of storage and transportation system. I will do one specific lecture on GMP, good manufacturing productions, and also during for the process end product, and also the entire production line for storage and transportation system later on another lecture. And at the end, the MI product that has been developed should possess this characteristic. For example, it has high rhizosphere competence. Rhizosphere as it can live very well, thrive vitally in the root area, which has low oxygen concentration in the soil, and also it has a good compatibility with the indigenous, the naturally occurring rhizobacteria within the soil. In addition to that, we also have to make sure that the whatever plant promoting plant growth criteria that inspired by the microbial inoculant by various mechanisms, for example, solubilizing the phosphate or potassium in the soil, or nitrogen fixing. This is called various mechanism. By segregation of biological active substance, which are normally secondary metabolite, growth hormone like auxin and so on. So they must have certain mechanism that has been identified in order for it to be patented as a biofertilizer. Okay, and then this culture is also easy to multiply, meaning that you can use inexpensive and easily available medium. Typically is the complex medium such as molasses to grow the biomass to high concentration or density. Of course, sometimes we can also use chemically defined media to facilitate the modification of the ratio of substance influencing the proliferation of microbial strains. So if we use the complex medium, which is cheaper, if the density is not so good, then only we will use the chemically defined. We will optimize different defined medium. Once we use defined medium, usually it's more expensive lah. But if we wanted to have a good density, we might still consider. Okay, then there will be a range of optimization experimentation to choose cheap defined carbon culture, carbon source, nitrogen source, and so on, that will maximize our biomass. Okay, and also the MI product should also exhibit wide scope of action, meaning that let's say it is good to grow fruity fruits, for example chili, tomato. At the same time, it can also be good to grow paddy, for example. So then the application, the scope, it is suitable for a wide range of the plant is desirable. Of course, it has no threat to natural environment, like the example I told just now, that its presence cannot wipe out the rest of other naturally occurring beneficiary microbe. So this is often quite tricky. It sometimes need long-term monitoring, testing. So to come up with a new MI is not always easy. Usually in Malaysia, we just follow whatever that has been developed by overseas, US, Australia, Europe, then we will. Okay. So this is an important slide to tell you, you should be able to name at least up to around 7 different type of the microbial count, and you need to know this CFU term, colony forming unit. So this is basically when we grow the MI culture, we will do a series of dilution to see distinctive colony in the petri dish, and we will count how many cell is grown in the culture by time multiplied by the dilution rate. Okay, so for example, rhizobium, they are the fast growing type, slow growing type. Nitrogen fixing bacteria, very common one. Phosphorus solubilizing bacteria, based on solubilizing organic P or inorganic P. In fact, for nitrogen also, they are the inorganic and organic portion of the nitrogen conjugates. to measure nitrogen, there are at least around 11 type of different format of organic and inorganic. So this for people who do detailed chemistry work, they will go into the detail. But as an engineer, we just calculate the nitrogen as a total nitrogen, including all the organic and inorganic. But in terms of formulation, for P, they are this organic phosphorus. There's also silicate solubilizing bacteria, because silicate is insoluble, is a kind of is a kind of mineral. Some bacteria can also solubilize silicate. Silicate is usually a major component of sand. In a sandy area, after solubilized, the silicate can also give other organic compound like silicon oxide and so on, that can be taken as a carbon source. Multi-strain consortium, meaning that the MI, either we develop into the 6 type of the specific group of the strain, or we can put a few different strains from 1 to 6 into the multi-strain culture. And usually this is more challenging, because there may be antagonistic effect among the multi-strain. They might be fighting for the carbon source and nitrogen, and causing some strain to lose their colony count halfway of the growth curve. Okay, so for liquid fertilizer, biofertilizer, ideally you must have at least 0.5 to the power of 9 colony count to 1.5. For solid, you can have a lower 0.1 to 0.3 to the power of 9. Okay, and minimum shelf life depending on different country, but typically it is around 6 months. Okay, of course different country will have different standard. If we look at the Indian standard, they will take into consideration the physical form. You must follow certain form, for example granule, and then you must have the minimum count of the viable cell, as I said in CFU here. The level of contamination, for example, you may have lead or mercury in your soil or compost. You must keep the level below the acceptable concentration. And also in the Indian standard, they decide if you use any carrier, the particle size must be less than certain size. The water content probably cannot be more than 40%. If it is more than 40%, it's like you are buying water only, isn't it? Then also this is referring to solid MI. Yeah, too wet is also not good for keeping the microbial stable. And they also set what is a common pH, usually around 6 to 8, to make it stabilize. And also the efficiency characteristic, which is this one. Yeah, colony count is the efficiency characteristic. And they also, in the Indian standard, they also specify if you want to call it as a biofertilizer, microbial inoculant. These are the one that has been patented: Rhizobium species, Azotobacter, Azospirillum, phosphate solubilizing bacteria. These are the 4 group, and they must follow this characteristic. This one is the general one in the combining US, Europe at large, but this is specifically for India's case study. Okay, and if there is a mycorrhizal fungi, which is very common in the biofertilizer, you must have 100 viable propagules. This is also CFU, colony forming, per gram of AMF. Okay, propagule, I think this is, this kind of fungi, they are like in a bunch globule form, so you must have 100 viable according to the method that's seen under microscopic, you must see at least 100 to classify it as a qualified mycorrhiza fungi biofertilizer. Okay, so as a general guideline for quality assurance, these are the 8 principles. Amount of living cell in CFU, carbon content, you must have organic matter of MATTER, greater than 20%, because in the organic matter you will have humic acid to becoming a housing to keep the microorganism or absorb the microorganism. The water content, it will varies with country, but usually it's around 40% or less. It cannot be too low. If too low, the microbe will die. If too wet, the microbe doesn't have enough oxygen. So the pH, the size of the carrier, pH may varies a little bit depending on strain of the microbe. For example, if it is lactobacillus, it may prefer acidic pH, but most of the microbe are good in around 6.5 to 8 pH. The size of the carrier is controlled. Yeah, you cannot have too big. If too big, the absorption surface is not so optimum. Appearance means it cannot be too smelly or too dark color, or the color also have control. Contamination is both pathogen for the biohazard and as well as heavy metal, for example lead, plumber, and so on. And of course the expiry date of the MI, which is in China, it has to be greater than 6 months, at least 6 months, 6 to 12 months to be qualified as a biofertilizer. Okay. Summarizing the MI, so these are the 5 major groups that you must be able to articulate. PGPB is one of this strain called plant growth promoting bacteria. Some of these nitrogen fixing, they are fixing, some of these rhizobia, they are fixing nitrogen, some they are not fixing nitrogen, but still they are providing some plant promoting mechanism. AMF, I told you already, it must have certain globular structure of colony. And phosphate solubilizing, for your information, phosphate is the major element for all metabolism. In the metabolism, when we digest the glucose, we go through TCA cycle. It is a series of phosphorylation process that need phosphate to generate ATP, which is the energy source in cell to promote growth and cell duplication. Yeah. So inside the rhizosphere here, or within the interspace within the root, where all the microbiota, microbiota means a range of different strain of microorganism colonize in a particular site and form a habitat that play a role in promoting plant growth. One of the strain inside the microbiota is called rhizosphere, rhizobia, live within the space of the root. They will mainly to, for example, degrade the soil organic matter. So the microbe will excrete or segregate the exoenzyme. Yeah. Okay, this microbiota, it releases the exoenzyme which degrade the soil organic matter and release the soluble C and N and other nutrients, yeah, and amino acid or organic acid. Okay, and then there is also forming from the root, they will also release some organic compound and ions. Yeah, so this will synthesize more other carbon-based and nitrogen-based nutrient to be uptaken by the root. Okay, at the same time, this exoenzyme is also degrading a range of different type of soil organic matter in the compost. Okay, so this symbiosis process all attributes to the contribution by the microbiota. Okay, for example, this is your MI. Your MI is coating with the clay. Yeah, so this there are a lot of interrelation actually. Okay, and the MI attached on the clay, it will be more stabilized, yeah, as compared to the naturally occurring microbiota. It can be leached through the rainwater into the soil, but if it is attached into clay, it will sustain in the soil for a longer time. So both of these, either your immobilized MI or the microbiota, all will degrade the soil organic matter and provide the soluble nutrient of C and N to be uptaken by the root. And all this synergistic effect, for example, azospirillum and the phosphate solubilizing bacteria inoculation can enhance the biometric, nutritional, and yield of the pearl millet. Pearl millet is a kind of wheat grain crop. Yeah, it's reported in this journal paper. Biometric means the application of statistical analysis of biological data, yeah, including what kind of strain is produced after this inoculation, what kind of primary and secondary metabolite is produced. These are all under biometric. And nutritional value is usually referred to NPK, macronutrient, and also micronutrition increase in the content. Okay, yield, of course, more crop yield will be observed. And then when they compare against the control, it is found that the one inoculated with this mixed azospirillum and PSB has increased the dry weight of the root and the shoots at 75 days after sowing. Okay, so it is observed that the availability of the P, as I said, phosphorus is not so solubilized in soil, but with this azospirillum, it increased the soluble and which is available to the plant. That's why it's called available P by this PSM, yeah, because this phosphorus solubilizing bacteria tends to solubilize the inorganic or organic P in the soil. And then azospirillum can increase the nitrogen fixation. Yeah, it's a nitrifying bacteria in the rhizosphere. So both of these increase of N and P exerts the synergistic effect and causing the plant to grow better. Okay, and then it was found that both of these azospirillum and PSM were isolated from the mangrove. And mangrove is a kind of a natural forest by the seaside. Yeah, they have a mixture of seawater as well as the inland plain water. Yeah, then the microbe is versatile in giving a lot of good enzyme in terms of nitrogen fixing and solubilizing. So for example, we look into how does the rhizobacteria look like. One of the common example is azospirillum. This is a strain, genera, and Lipoferum is a species. Yeah? So this is genus. This is A stands for azospirillum genus, and this Lipoferum is a species. So as long as we saw rhizo, this is root associated bacterial that forms symbiotic relationship with the plant. So in the exam, you must be able to elaborate what kind of the synergistic effect of bacteria and plant. For example, nitrogen fixing bacterium, Pilobacterium species is a gradually growing nitrogen fixing, and a fast growing is a PSM B bacterium licheniform. Both were isolated from the rhizosphere of mangrove. Mangrove, you know, there has a lot of this root that some of it is exposed in the air, so a lot of the microbe like to live in this mangrove. Because they are highly versatile, they can thrive even with seawater, or when the tide comes up and the tide goes away. When the root is exposed in the air, they can still thrive. They can live very well. As in this journal, there will be a series of journal paper that you can do further read. Okay, so this is how it looks like. arbuscular mycorrhiza fungi. So this is a root, the yellow color, and a lot of this hairy structure is how the AMF looks like. So it's a very powerful fungi with a lot of tiny hairy structures that release a lot of this lignocellulosic enzyme to degrade organic matter efficiently due to this highly tiny hairy nanostructure. So we like to call it as symbiotic fungi, which penetrate the cortical cells of the root. This is the outer layer of the root of a vascular plant forming arboculous tubes. Yeah, see, it can even penetrate certain part of the root to make the transfer of the nutrient from the soil to the plant much more efficient. For example, phosphorus, sulfur, nitrogen, and micronutrients including magnesium, boron, calcium, ferrum, and so on from the soil into the plant. Okay, usually they help the initial colonization of the microbe onto the plant, and this will slowly evolve into the formation of vascular plants. It has kind of texture that have a vascular type. Yeah. And another type is this famous nitrogen fixing bacteria, very famous. It's called azotobacter. It was told that in one of the MI formulation is using VAN, yeah, and also azotobacter and PSB, yeah. This was reported to enhance the phosphatase activity, metabolism, to break down organic matter, either sugar-based, lipid-based, or protein-based, to enhance the soil nutrient, yeah, nitrogen, phosphorus, potassium, cuprum, zinc, ferrum. This one I think is calcium. This is calcium, Ca, and manganese. Yeah? So it has been reported to enhance the growth of this plant called Stevia baudiana plant. Stevia is an artificial, natural sweet sugar substitute, in which it is also a rhizosphere, in the rhizosphere area, in the root area of this plant. Okay, so when used in three together, it has shown positive effect for this MI successfully applied on alkaline soil and also enhance acid phosphatic activity. Okay, so in this journal, so there are many more of example of MI. Some is free, some is consortium. Even the free living without any immobilization, all these common strain can be used as a MI, but of course they don't have a good shelf life if not formulated. We call it as free living. Okay, just spray directly, and then it has a short shelf life of a few days. Unless we immobilize or encapsulate, it can stand for several weeks within the housing carrier. Symbiotic effect of Rhizobium frankia and Annabella azorea. So these are all different type of strain. Azospirillum, just now we mentioned. P stabilizing some of these fungi which produce penicillium species. Automatically this will give the plant higher resistant against pathogen or disease because of this antibiotic functionality. AMC, right? And so on. This arbuscular mycorrhiza is a fungi, yeah. So very famous for mobilizing phosphorus. It can also be combined with other species here. These are all known combination of different type of the species, Lecaria, 1, 2, 3, 4, and 1 on 2, which has been successfully researched and showing that they can live well together to be formulated as a combined microbial inoculant, so they don't have they would not have an obvious antagonistic effect for the competition of food in terms of carbon source and nitrogen source. Okay. This one in other source, in other source of journals on page 48. This one there will be an article later on in the list of references. In this book chapter, it shows that 1, 2, 3, 4, 5, 6, 7 type of function of the MI, nitrogen fixing, phosphorus, solubilizing, plant growth promoting. This is a very big group, including Rhodosunomonas and many types, lactobacillus also. Potassium solubilizing, yes. Sometimes they don't put solubilizing, but we know these two macronutrient, phosphorus and potassium, are not soluble in water, so they must need some microbe to solubilize it. Then only it can solubilize in water to be uptaken by the plant's root. Occasionally, there are also formulation on zinc solubilizing or sulfur solubilizing or silicate, just now I have mentioned, because all of these minerals exist as a rock in the soil and usually are not solubilizing readily and require this microbe to do the work. Okay, so I will give you supplementary document to read. Okay. For the impact of microbial inoculant on soil microorganisms, there are a range of research work that can be conducted using high resolution equipment such as denaturing gradient gel electrophoresis or RFLP, terminal restriction fragment length polymorphism, or DNA restriction analysis. That these are a range of the name of the analysis that can be analyzed, used to analyze the microbial community or microbiota in the biofertilizer, using this powerful instrumentation called biolog. Okay, this G is together, biolog, and the plate, it has a lot of automatic arm, robotic arm to do this kind of PCR and DNA screening. Then in a short while, we can understand what are the strain contained in the microbial inoculant. But this equipment is very expensive. It's around $500,000 ringgit Malaysia. Okay. So to understand the impact, we can use this biolog to understand whether it is caused by certain enzyme. Yeah, we can use DNA restriction analysis to identify specific type of enzyme or protein or plant promoting hormone and so on, using a range of different type of analysis. Okay. And of course, at the end, it is we want to make business from bioprocessing, right? So we want to know whether our MI product, the microbial inoculant, can be sold at a good price. When it is sold, we must make sure that it has some physiological active state. Although I said it is formulated in the hibernated stage, but most importantly, when diluted with nutrient or water or any solvent, it can go back to the physiological active state to exert the effect, for example, to excrete protein, hormone, and so on. It must also show that it is fully functional at all environmental conditions. For example, if you sell it in China with 4 seasons, preferably it can be used for all 4 seasons. Or if in Malaysia the weather is very hot and humid, this also should be suitable for the environment in a specific country when it was sold. So of course, it need to have an enhanced shelf-life, viability in terms of CFU, colony forming unit, lasting for a long time. Of course, importantly, the result has to be consistent in the field performance, not the effect of different temperature or weather will be minimum to effect on the consistency in the field performance. And reduce phytotoxicity, not causing other naturally occurring microbe to die. Yeah? So most of these content are extracted from this paper, Castro-Sowinski 2016. So in fact, in terms of the MI product, it has a huge global market, but 80% of this MI is monopoly by Rhizobial, followed by 50% of phosphate solubilizing biofertilizer. Yes, nitrogen is very important. Rhizobium is also closely related to nitrifying. Yeah? And then others, around 6-7% is the formulation of this mycorrhiza fungi. And back in year 2017, this is a business case of 1.26 billion USD. And you can imagine now that a lot of the fertilizer, chemical fertilizer, the price has increased 50% and above. Malaysian ringgit also appreciate around 30% following COVID and so on. So it's making, and for your information, Malaysia import around 80% of the fertilizer from overseas, from the US, Canada, Australia, and so on. So if we can convert food waste or any biomass to organic fertilizer, it can represent a huge economic impact. So estimatedly, this will escalate to 2.3 billion business case worldwide. And in fact, within these 5 years, the growth of the biofertilizer demand is about 10%. Okay, so as a biochemical engineer, you should be able to name some of these common microbes, especially the genus Azotobacter or Azospirillum. This is bacteria and this is fungal. So you should be able to differentiate what are bacteria, what are fungi, and some of these commercial name of the biofertilizer. Of course, this I don't expect you to remember, but across different continents, there are different type of the microbe. And then when you see this name, you know this is consortium, the mixed culture. And if you see one name is a pure culture, so this is mainly for nitrogen fixing. And then there is also continuously more mixed type of the some of them even mix more than 30 type. So there's a pro or con of using pure or complex. If it is complex, meaning that it is capable of degrading more nutrient, more rocks, different type of rocks, but their individual performance may not be as good as the pure culture. Okay, so they have all the pro and con when used pure or mixed culture. Okay, as a conclusion, biofertilizer is a supplementary, renewable and eco-friendly source of, can provide this kind of eco-friendly source of plant nutrients to the soil and promote plant growth. Moreover, if we valorize the waste to biofertilizer as a product, this is highly desirable because we can reduce the organic waste at the landfill sites. Okay, and we can promote integrated plant nutrient system, because when you buy pure chemical fertilizer, it may not be, the nutrient may not be as complete as the biofertilizer, which also supplement those small micronutrients like magnesium, boron, and so on. So biofertilizer is very vital to support sustainable farming practices. It can also mitigate any pitfalls of the growing demand of global population in food and reduce the widespread of chemicalization of agro ecosystem. And when we use any microorganism to rise within the rhizosphere, this can has a significant multifarious plant growth promoting activities. Yeah? However, relatively, although we know 80% is rhizobia type, but mainly they are used as a free living microorganism. Not too many have been formulated in the form of biofertilizer with prolonged shelf life. This is due to formulation costs. Yeah, okay. So as a biochemical engineer, yeah, if you become an entrepreneur, you should be more activated to look into this market and collaborate with a UTM professor doing research. The company can expand the application to attain the objective of organic farming in terms of functionality, sustainability, activity, but all in all, the cost efficiency is also important. That's why we need to understand to where we can get cheaper carrier that can provide high activity to our end product. It is not always easy, like even sometimes to develop a highly effective with a low cost can easily take 5 to 10 years and so. Okay, so with that, that will conclude the lecture for development